Hi, welcome. Hi. How are we doing tonight? Okay, so you were saying you've got your mock coming up, where you've got one, remind me, one, two, four, and six was what you said, wasn't it? With regards to your mocks. So is there anything specific in those topic areas that you are really struggling with? Anything you want me to cover specifically? And I'll start doing it. Hi guys, welcome if you're joining me. I thought I'd go live tonight and start doing some actual revision because I know some people have mocks coming up. So obviously, if you are in year 12, can you please go through mitosis and meiosis? Absolutely, of course I can. So when you're looking at mitosis, and meiosis. So if we're going to compare the two, meiosis, I always have to sound it out so I know what it is that I'm actually trying to write because my spelling is atrocious sometimes. So if we're looking at mitosis and meiosis, one of the first things I would do is I would start blurting out bits of information that I already know and then try and fill some gaps. Now, I am all about drawing things. I absolutely love drawing things. I'm not very good at it. My writing is atrocious, but that's fine because I'm not an artist. I'm not trying to be an artist. I'm a biologist. And so, therefore, what I want to do is I want to make sure I know the difference between the two, but consolidate it down to the minimum part of it. So, mitosis is about getting genetically identical cells. So straight away, if I'm going to talk about mitosis, I want to talk about genetically identical cells. If I'm going to talk about meiosis, then I want to be talking about the fact that they are going to be genetically different. And the reason they're genetically different is because variation will potentially occur. So straight away, I'm just trying to think about where a question might go. Um, Okay, so if we are looking at this, let me just actually add in. What do I want to add in? I don't even know if I can add it in here. I'm not allowed to disclose that I'm elsewhere. Um, Mitosis then. Next thing I'm going to do is I always want to talk about my PMAT. So you should know PMAT when it comes to mitosis. Now, when we look at meiosis, I've still got PMAT, and I'll talk through what each of those are. But PMAT happens twice. So I've got prophase, metaphase, anaphase, telophase. Hey, Miss, just wanted to say that thanks to you, I got way above. Oh, that's amazing, Amelia. I'm so proud of you. Well done. That's really, really amazing news. Well done. Keep going. It's exciting that it's like got you where you want to be, and now it's the starting point of where you're going to move forward. So that is awesome. You should be proud of yourself. All that hard work finally paid off. Okay, back to this PMAT. So whenever I talk about mitosis or I'm talking about meiosis, I always straight away go PMAT. PMAT 1 and PMAT 2 when it's meiosis, just PMAT when it is mitosis. So we have got prophase, we have got metaphase, we have got anaphase, and we have telophase. So Why is it that we have PMAT, prophase, metaphase, anaphase, telophase, just once when it is mitosis, but then if we're looking at it with meiosis, we've got prophase, metaphase, anaphase, telophase 1, prophase, metaphase, anaphase, telophase 2. Now, one of the things I will say to you is when I'm abbreviating, like I'm writing down PMAT, or if I am abbreviating in my note form and I'm writing, for example, concentration, CONC dot. I always abbreviate whenever I'm writing because I'm lazy. I don't want to write it all. I want to write what do I need to flag my brain to put that information down in the exam. And so therefore, one of the warnings I would say for you from reviewing papers today is don't abbreviate in the exam. Still, students abbreviate, and because they abbreviate, they lose out on marking points. If you're going to write concentration, write it as concentration. So if I'm writing PMAT to prompt my brain to give me the information forward that I want, I need to write in the actual exam prophase, metaphase, anaphase, telophase. Hi, Alex. Nice to see you. Okay, so let's keep going with this then. So if I'm going to be looking at my mitosis versus my meiosis, again, I've got my prophase, metaphase, anaphase, telophase, 1, prophase, metaphase, anaphase, telophase 2, if it's meiosis. You'll be fine, Alex. Don't worry. Module 4 is not that difficult. Although it feels like it is, it's actually not. I'm 2 days into A-level biology, but I love it at GCSE, but it's overwhelming. Imogen, I'm really sorry to say this to you, but it's going to get more and more overwhelming. So are you doing OCRA? The one thing I would say to you is start as you mean to go on, get going, because if you don't, I will go over module 4 after this. I will, don't worry. Because I was on earlier on, and so I'm going to start with what she wanted, and then I will come to the next bit for you guys afterwards, okay? I'm just getting up my resources. It is a heavy module, module 4. But going back to you, Imogen, it is heavy going. It is hard, but keep going with it. It will make a big difference by you persevering and starting as you mean to go on. So I'm just going to do a quick whiz through for the prophase, metaphase, anaphase, telophase side of things. This is going to work out, actually. Let me just... And I want to switch my screens. Let me just switch screens right now. Okay, so I'm now going to switch screens and I'm going to talk about the cell cycle. I'm going to go through mitosis. I will cover, Alex, I promise you, after I've covered this, I will go and do some module 4 for you, okay? So the cell cycle you should know, interphase, G1, growth, protein synthesis, S phase, DNA replication, G2, growth. That's all interphase. Now, that is not mitosis. Do not get confused. Mitosis is not the same as the full cell cycle. So the full cell cycle is interphase, and then we've got mitosis, which is our prophase, metaphase, anaphase, telophase, and then cytokinesis. So mitosis is really important when we're looking at growth, tissue repair, and asexual reproduction. Now, that again is important. Asexual reproduction, because it is about getting that genetically identical daughter cells. Oh, thank you for the gifts, by the way. For those of you who are gifting, thank you for that. So yeah, so asexual reproduction, mitosis, we're going to end up with genetically identical daughter cells. And again, it's important that you get that repetition in your head of those key phrases. If they're ever asking about mitosis and you haven't used the word genetically identical, but you've just said daughter cells, you're not going to be getting the marking point. So do make sure that you try and give as much of those key terms as you go. So mitosis is our prophase, metaphase, anaphase, telophase, as I've said. Now, Another thing to note, for those of you who are year 12, you won't have gone into this detail yet, but for those of you who are year 13 and who have started already module 6, you should know about genes, and you should know that genes are going to be used at causing the expression to occur, or they could be used to inhibit the expression. So it's important that we know that we actually have genes that are going to stimulate for cells to go through cell division. Now, when the cells differentiate, they've already gone through that process of mitosis to give us two daughter cells. When they are going through differentiation, they are no longer able to go through mitotic division. So be aware, whenever we talk about mitosis, what we're saying is we're producing genetically identical daughter cells, and that is controlled by genes. I always go live on a Wednesday, and I've started again tonight, and I will keep going when I can. With regards to these, my actual group sessions are going to also be on a Wednesday, but I'm also going to offer them on a Monday. I just haven't advertised that one yet. Hi, Chidima. So going back, we have genes which are going to stimulate cell division, but we also have genes which are going to suppress it, because we don't want too many cells in an area too quickly. We don't want tumors to form. And therefore, because of that, we need to suppress the genes. So remember, genes can either stimulate you want the transcription to occur, or we might inhibit transcription to occur. So therefore, what we're saying is we're going to have expression of proteins that are going to occur that will therefore cause cell division to occur, or we're going to have the expression of proteins which are going to suppress the cell division from occurring. So one of the key things to know when it comes to module 6 is that there is a regulation which is going to cause either cell division or it's going to suppress it. And when a cell differentiates, it's no longer able to go through the process of mitosis. Time zone, I'm in the UK, I'm in Cambridge. So I do, what time zone is that? I don't actually know off the top of my head. I want to say time zone 1, but I might be completely wrong on that one. But it will be 8:30 UK time. Okay, carrying on with this, mitosis then. S phase is about DNA replication. So that was in interphase, that wasn't mitosis. But it's important that we note that the DNA is replicated, semi-conservative replication. So remember, that's your module 2, looking at your replication, one original strand, one new strand in every chromosome. So what we have is that's going to result in our chromosomes being made up of sister chromatids. And because they're being made up of sister chromatids, what we can now see is we now see that they are connected and there are two parts to it. So we always draw them as if they're X's, and the green part, I did this with Play-Doh when I was looking at my kids' Play-Doh. The green parts are showing you the centromeres. Now, the centromere is where the sister chromatids are going to be held together, and that's important when we talk about mitosis. On the original chromosomes, you'll see the green bits at the top and the bottom as well. Now, that's to show you your telomeres. And again, for those of you who are year 13, that's important when we're talking about our DNA profiling and our variable number of tandem repeats. So that's a module 6 thing that's going to come through. So you have mitosis that's occurring where you have these chromosomes have been replicated, but for mitosis to occur, we are going to start, remember, at our PMAT, prophase. Prophase, hi Effie, is where our nuclear envelope and our nucleolus is going to actually disappear. So if you've got a B and AS biology, what I would say is It's not possible to get the A star. You're absolutely on the right tracks, but you're going to have to work on your application skills. Because if you're only getting a B currently at AS, potentially your application skills aren't up to scratch and your details not there in your answers. So I would suggest if you are only getting AS B level, then what you need to do is really work on understand that content, but knowing how to apply it to the random situation. Right, back to this. Nuclear envelope disappears and the nucleolus disappears. So the nuclear envelope needs to disappear and the nucleolus needs to disappear because that DNA is no longer going to be contained. We now want it to be able to be separated because that's what we're splitting up. Mitosis is about the nuclear division. We're going to divide so that we end up with two genetically identical daughter cells. And so what we're going to do is we're not only going to get rid of the nuclear envelope, not only get rid of the nucleolus, but also we're going to condense those chromosomes. So for those of you, again, I'm going to jump to year 13, module 6 right now. The condensing of the chromosomes, the supercoiling, as we call it, the supercoiling of the chromosomes, when they condense down into a really compact place, you should know, and this is where I look for things around me that can help me show this. This one will do. So here what we've got, I'm going to use a wire. So if this wire is showing you my DNA strand, you should know eukaryotic organisms have histone proteins. And you should also know that DNA is wrapped around those histone proteins twice. And so if I wrap around my DNA around my histone protein with this hand, hold on one second. There we go. So I now have my nucleosome. Now my nucleosomes are a way in which I can actually regulate gene expression. So year 13, module 6, gene expression can be regulated with the nucleosomes. This is at a transcriptional level. What you need to know for mitosis is the fact that these two are going to condense down, which means they're going to come closer together. Now, these are my two hands, but within your chromosomes, you are going to have millions of these nucleosomes. So when you're looking at the nucleosome and you are looking at the supercoiling, it's basically pulling them close together. And what you have to understand is DNA is negatively charged. My histone proteins are positively charged, so they are attracted to each other. And therefore, that's why they stick together. And when we get them to coil closer and pack into a smaller condensed version, we are changing the way that they are interacting. And so that is important when you're looking at regulation. Nucleoplasm, plasm is just talking about a liquid. Nucleoplasm would be the liquid inside the nucleus. Excretion and ecosystems in 3 days. Check out, if you haven't already, Yusuf, go to my YouTube channel and watch the videos there would be my suggestion over the next 3 days, but only the content that you don't actually like or know. You will find it on here, but it'll be harder to find, whereas the playlist on the other side. What about the skin neuron thing? Do we need to know that? What about the skin neuron thing? What do you mean the skin neuron thing? Are you talking about receptors in your skin? And so therefore, are you talking about, for example, your mechanoreceptors or your thermoreceptors? I'm not sure what you're asking, Lambo. But back to this. So as you're going through mitosis and you're looking at what's happening, it's important that for those of you in year 13, you realize that content's going to come through in all that you've been doing. So for those of you who are currently year 12, it is going to feel overwhelming because right now you are slowly getting jigsaw pieces together. When you get to year 13, what should start to happen is you should have part of that jigsaw puzzle together and it should start to form a picture, but with missing pieces, and you have to fill those missing pieces. Do not memorize all the content. It's not going to work for you, and regurgitating isn't going to work for you either. So carrying on, metaphase, you can see lining up here. We have got my spindle fibers have moved, they've pushed and pulled the chromosomes to the center. And that therefore means they're now lined up at the equator. Because they're lined up at the equator, that is called the metaphase plate. That is where we have metaphase. So you can quite clearly see these lined up at this point here. Now I'm going to show you this bottom picture in comparison to this one that's up here. And the reason, I'm hoping you can see that, the reason that it's doing that is because what you've actually got is the difference between metaphase lining up at the equator and anaphase, where you have that your centromeres are going to be separated. And as the centromeres are being separated, those sister chromatids are going to be pulled apart. And because those sister chromatids are going to be pulled apart, what that's going to do is that is going to take one sister chromatid to one pole, the other sister chromatid to another pole. Now I'm going to take a step back a second and say to you that the spindle fibers, the proteins that are being produced by the centrioles, these spindle fibers that are coming out, are actually part of your cytoskeleton. And they're useful. And what they will do is they will produce them and then retract them back in. So the centrioles have moved to the poles. They have attached, by producing these proteins, made these spindle fibers, attached them to the centromeres, moved them to that position in the middle, and then they have pulled those sister chromatids apart. So what we're ending up with is, oh, I've hit the wrong button there. Hold on a second. We're ending up with them being pulled to those poles, and once they have reached, and only once they have reached the poles, have they then got to telophase. So you can see this V shape, which is really important on the during anaphase. One second, let me just go back a second. You can see this V shape on the chromatids as they're being pulled. Because those spindle fibers are retracting, you always get that shape. And if I go back to, no, I still want this picture. You can see the kind of V shape. Now, they have in the past asked you to draw that. So just be aware they could ask you to draw that. The other thing to note is telophase. We can see telophase because we're actually starting to get this less visible chromosomes as we had before. So during telophase, what we've got is those chromatids have reached the poles, and we're now calling them chromosomes. And another thing that students get confused at, it's a chromosome when it starts. When it has been replicated, it's still a chromosome, but it's a chromosome made up of sister chromatids. Anaphase, we're pulling those chromatids apart, and that's therefore going to result in, when they reach the poles, them being called chromosomes again. So chromosomes, chromosome made up of sister chromatids, chromatids pulled apart, chromosomes when we're at telophase. The nuclear envelope is going to reform. We're then going to have the chromosomes that are going to start to uncoil, and the nucleus is going to be formed. And then you need to split the cell. And that's where cytokinesis is going to occur. Cytokinesis, we're going to split everything evenly. We've now got two genetically identical cells. We just need to make sure that we pull in the membrane and completely separate them. And that's where your cytokinesis comes in, because you've got the fact that this cleavage furrow that's happening in the animal cells, and you have the cell plate. So you've got these Golgi vesicles that are going to line, and what they're doing is they're starting to produce the cell wall, because remember, plant cells have the cell wall, and that cell wall needs to be there to ensure that the structure of the plant cell is intact. So one of the things that makes it different for the animal cells to the plant cells, a way in which you can classify. So for those of you who were talking about module 4, this is something that a lot of students for some reason really struggle with. If you are looking at identifying between which of the kingdoms they come into, is it Animalia, is it Plantae, is it Protoctista, is it Fungi, is it Prokaryote? If you're trying to distinguish that, you need to be looking at some key parts. For example, is there a cell wall? If there's no cell wall, absolutely, we are looking at an animal cell. If it sometimes has a cell wall, but sometimes doesn't have a cell wall, then that could indicate that it's a protoctista. But you have to look further at the information they've given you. But if it has a cell wall, like a plant cell, is that cell wall made up of cellulose? And that's the bit that a lot of students, for some reason, don't pick up on. If they don't state that there's a cell wall present and that cell wall is made up of not chitin, fungi, it's made up of cellulose, it's a plant, and that would be a distinguishing feature. But what if they tell you that it's got a cell wall and it's actually made up of peptidoglycan? Then it's a prokaryote. So these are really important little tidbits that will be in the exam that you have to look out for, because if you don't and you see a structure, for example, that has chloroplast, you could make the assumption that it's a plant cell, but it could actually be an autotrophic protista. So just be very careful with that module 4. That module 4 is mostly about application skills to random situations. So the specific content you have to know, absolutely. When we're talking about the immune system, for example, when we're talking about the communicable diseases. However, what they love to throw in there is the bits about evolution, classification, and you bringing in GCSE knowledge along with your A-level knowledge that you have. So don't miss the extra bits, but also that will, this is basically, that's my skill. I would say that's what kind of makes me stand out to other videos that are out there when I do these lives. These live sessions, as you'll all know, they're not planned ahead of time. They're literally, I don't do these. Yes, my group tuition sessions, I absolutely do. And my one-to-ones, I absolutely do. I spend the time preparing them, marking them, etc. But for these sessions, these are about you being able to ask questions, and I will just go with wherever you take the path. And that's the problem that most students don't see, that it's the bigger picture with biology. You have to be able to see that final piece, those interlocking parts, because if you can't, this topic, or not even this topic, this whole A level is rough. It is so unmanageable because there's just too much. So it's important that you try to think of where there is biology in everything that you're doing and what they're giving you, because you will find it if you know where to look. Let you know I got old. Oh my God, that's amazing. Well done. That is really amazing. Congratulations on all the distinctions. Sorry, I'm trying to make sure that I do cover everybody, the different content that you guys want, and also remind it being there for the new students. But that's amazing. I'm really chuffed for you. Right, let me switch it up. I'm quickly gonna go for some. You've also got to excuse me. I've got a cold, so I'm trying to do this whilst I sound rough. No, I don't want to do that. I don't want to do that. Just gonna view time there. Right, let's look at meiosis quickly, and then I will switch across to the next one, which is module 4. Alex, if you're still here, which specific part of module 4 are you looking for, by the way? And I will try and make sure I cover that bit first. Okay, so meiosis then, just to go through meiosis, I said before, you have your prophase, metaphase, anaphase, telophase 1, prophase, metaphase, anaphase, telophase 2. Now, you'll notice anyone who comes to my sessions, anyone who watches my videos, you'll notice that I do a lot of repetition. I say the things again and again and again and again. What you have to realize is you need to be doing that repetitiveness to really ingrain it in your brain so that you can then pull it forward when it comes to the exam. So yes, there are videos where I'm going through questions and I'm answering them, but one of the key things I would say is if you've watched a video that you have feel like you've got, don't think you shouldn't watch it again. I know fine well some of my students will leave my lessons and be like, oh yeah, I know it now because Christine taught it to me and we went through it and it's really good. I still would retest them again next session to check, did they actually remember what we did last lesson? I'll do it in a month's time. I'll do it in 3 months' time. So for those students who've been working with me for the last year, they know that I will ask them questions on any topic area because they should always be repeating that content in their head, keeping it fresh. And that's one of the things that AIQ chat was actually built for, so that you can do a small but constant flurry of questions on lots of different topic areas. Anyway, back to this. So meiosis then, prophase 1, well, we know, as we just discussed, nuclear envelope disappears, nucleolus, chromosomes condense, centrioles move to the poles, produce those spindle fibers. Now, this is the bit that's different. So if I was making my notes about mitosis, meiosis, my PMAT doesn't change apart from just a slight few extra bits. So prophase 1, homologous chromosomes are going to pair up. Now, homologous chromosomes, homo, same. Chromosomes have the same length, same genes at the same loci. And that's represented here with my blue, my red. So these are the same genes, but these are different versions of the same gene. So that's where module 6, for example, mutations will come in. What's happened? What is that mutation? Is that mutation a substitution? Is that mutation a insertion? Is that mutation a deletion? Whatever that mutation is, it has resulted in an allele, which is a different form of that gene, a different sequence of the DNA. If you've got a different sequence of the bases in your DNA in that gene, that could then potentially, and I'll say that potentially, code for a different amino acid sequence, therefore a different primary structure for your polypeptide. So it's important that when you are doing your revision notes that you are getting these little bits of, oh, that's important. Mitosis, PMAT, but meiosis, prophase 1, I've got these homologous chromosomes. The other thing to note with prophase 1, which is really important, is that crossing over can occur. It is the only time that crossing over can occur is prophase 1. Metaphase, we know metaphase in mitosis was about that lining up at the equator, exact same thing. However, slight variation because we're talking about meiosis, the homologous chromosomes are pairing up at the equator. Now, this is the important part when we look at independent assortment. So when you're doing module 6 and you're talking about Mendel's law, excuse me, of independent assortment, I talk so fast, I take in too much air, it makes me get the hiccups. So with meiosis, you have got this independent assortment. What do we mean by that? So independent assortment is about the orientation of those homologous pairs. Now, I've got different colors here. I've got dark purple, I've got pink, and you can see I've got short ones and I've got long ones. So if I have short ones, on the left side of my screen, as I'm looking at it, I have got the short purple, which has been lined up with the long pink. I have the short pink, which has been lined up with the long purple. Well, what happens if it had actually orientated itself the other way around? What happens if it had orientated itself and we had this bit of crossing over that occurred during prophase 1? When they are lined up at the equator, what's going to happen is once they're at that metaphase plate, once they've been lined up, they are then going to move on to anaphase 1. And during anaphase 1, they are going to be pulling those homologous chromosomes apart. So however they lined up at the equator, that independent assortment of each other is going to result in potential variation in the gametes that are produced, the daughter cells that are going to have half the genetic information. What kind of questions can come up based on meiosis? Lots of different questions. They could ask you to compare mitosis with meiosis. They could give you a diagram showing you the stages and ask you to explain what's happening. They could link it to the module 6 side of things and talking about the independent assortment and how it could result in a predicted ratio that isn't as expected. So it really depends what way they want to go. So if you are year 13, they can ask it in lots of different ways. If you're year 12, it will be more about the stages and how it results in either genetically identical cells, mitosis, genetically different cells that are haploid, if we're looking at meiosis. So let's just explain how we get those haploid cells. So again, anaphase 1, we've got the spindle fibers are going to retract into the centrioles. They're going to pull the chromosomes to the poles. Well, as they're pulling the chromosomes to the pole, listen to my wording, the homologous chromosomes are being separated. So this again is that little bit of difference between mitosis and meiosis. During anaphase in mitosis, sister chromatids are pulled to the poles, separated at the centromere. In meiosis and anaphase 1, homologous chromosomes are separated. Then we go through telophase, they reach the poles, we end up then going back to prophase 2. Then I'm just jumping to metaphase 2 because metaphase 2, we're lining up at the equator again. So we're not changing anything here. We're saying it's the same stuff that you had before, the same key content. The most important part is knowing which bit we're at. Are we at metaphase 1? We're lining the homologous chromosomes up, or are we at metaphase 2? And at metaphase 2, we're lining them up in those individual cells. And then obviously anaphase 2, this time it's my sister chromatids are being pulled apart. When I was in year 13 at normal. Oh, well done, off to university, exciting. And then telophase 2, chromatids reach the poles, and now chromosomes, nucleus reforms. So it's important to know when we're talking about meiosis, you had a diploid cell. The diploid cell went through DNA replication. You've now got your chromosomes made up of sister chromatids. So regardless of whether it's mitosis or whether it's meiosis, that's what we can say. And then what we have is we have got our meiosis 1 is all about those homologous chromosomes being separated during anaphase 1. So you can see here I now have 2 cells, and those 2 cells are half the original one. So this one was a diploid cell, 2N, 2 copies of every chromosome. Now I only have 1 copy of each chromosome. I have 1 short, 1 long. Here I had 2 shorts, 2 longs. So I've already reduced it, and then I'm going to separate the sister chromatids. Excuse me. And so that's where meiosis 2 comes in, and anaphase 2 about separating those sister chromatids so that we end up with these haploid daughter cells. And those haploid daughter cells could be genetically different because they may not have the same genes or alleles because of crossing over occurring. So, oh, that was a slide I put on before. So here you have it with my little diagrams here. Diploid, DNA replication, 2N sister chromatids. If we're talking about humans, 46 chromosomes, 23 pairs. 23 pairs means it's diploid. 1 to 2, or 1 to 2, 1 to 22 is your autosomes. Again, that's module 6. The 23rd pair is your sex chromosomes. So they could bring in module 6 at any point if they wanted to in this. What time do these sessions start? So these sessions that I'm doing right now will be starting at 8:30 with regards to the free lives that I do. My group tuition sessions are 7:30. I do them before. Right, so I'm going to go back now. I hope that's been helpful. Right, you can go away. So module 4, I don't know, Alex, if you are still there. And if you are, did you have a specific part of module 4 you wanted to cover? Or is it any part of it? Really hot. There's module 5 for you. My adrenaline is pumping, and therefore, because my adrenaline is pumping, I've got more blood flow going through. So my arterioles close to the skin have dilated, and that is resulting in the heat being radiated out from my skin. Okay, what's the difference between metaphase 1 and metaphase 2? So metaphase 1, whether take away the 1 or 2, first of all, what is metaphase? Metaphase is lining up at the equator. So if we're looking at metaphase 1, our homologous chromosomes have lined up at the equator together. If we're talking about metaphase 2, in metaphase 2, there is no homologous chromosomes anymore. It's just the chromosomes have lined up at the equator. So metaphase 1, homologous chromosomes are lined up at the equator. Metaphase 2, the chromosomes are lined up at the equator. You are very welcome. You are very welcome. So guys, if you do have anything specific on module 4 you want me to cover, please do just go, Miss. Can you also go over phagocytosis, like the steps when it's engulfed? Yes. Okay, give me a second. That's the sort of thing I'm looking for. If you can give me a specific example that you're struggling with, I will find a part of it and tell you more. Right, phagocytosis. Oh, excuse me. Right. Let's go here. Right, here we go. So when we are looking at phagocytosis, so here I have my phagocyte. Now a phagocyte, we know, is a specific type of white blood cell. It is involved in the non-specific immune response, and it's going to do phagocytosis, which is where it's going to engulf and digest. So that's GCSE level. It's a white blood cell, it's going to engulf and digest, and that engulf and digesting is going to hopefully get rid of this pathogen. Now, it doesn't matter what the pathogen is. It might be dust. It might be a bacterial cell. It could be a fungal spore. It could be a cell inside the body which has actually gone through apoptosis. Again, bringing in module 6 for you. So whatever it is that's going to need to be digested, the phagocyte is going to need to engulf it and take it inside. So module 2 is about you understanding you have different cells that are differentiated to have specific roles to play within the organism. So this phagocyte, its role is that it is going to engulf and digest. So therefore it has lysosomes, these organelles, these membrane-bound organelles inside the cell that are needed for it to do its function. Now, what did I say before? I said that the phagocyte is going to engulf and digest. So if it's going to engulf and digest, what do we use for digestion? We use enzymes. So what is going to be held inside those lysosomes? Those lysosomes are going to have digestive enzymes, hydrolytic enzymes that are going to break down whatever that debris, pathogen, etc. is. So if we're looking then at phagocytosis, what have we got occurring? Well, my phagocyte had to move. It moved to get to where there was that pathogen. In this case, the pathogen was the prokaryote. So this was a bacterial cell. I can tell that because it's really small. I can tell that because it has a flagellum. It's got circular DNA. It's got 70S ribosomes. It has a cell wall, which is made of peptidoglycan. So again, bringing that other content forward and not just, oh, Alex, you disappeared. And not just looking at, oh, I learned about cell structure over here. That's done now. It's going to potentially do this with it, okay? So be aware it's not standalone each module. There's so much overlap and interlinking that that's how you make it make sense. So now I'm going to take a step back again. And for those of you who are just joining me, or this is the first time you've come to my sessions, yes, I will jump around all over the place. And the reason I do that is because um, it's actually where my brain is going. So I've got ADHD, which means that I go off in tangents all the time, but also that I know is useful and helpful for me because it's how I can tell a story. And by being able to tell that story means I can make it make sense. So again, I'm going to take a step back and talk about the fact that we had these lysosomes, which are membrane-bound organelles that are going to contain these enzymes inside. Now, we know the cell itself has got a membrane. So we know that the outside of the cell, the phagocyte, which is a type of animal cell, has got a plasma membrane. So we know it's got a plasma membrane, which is made up of phospholipids, the phospholipid bilayer. We know these membrane-bound organelles, the lysosomes, have a membrane-bound membrane. Therefore, they also have a phospholipid bilayer. Important. So the cell is differentiated. It's got these lysosomes. The lysosomes have these membranes, those membranes which are containing those digestive enzymes inside until they're needed. So the first thing we need to talk about is, well, how does the phagocyte actually get there to wherever that pathogen is or that chemical? Well, it is actually attracted towards the pathogen by the chemicals that the pathogen is going to produce. What do I mean by membrane bound? Basically, there's a membrane surrounding it. So, hold on one second. If I go to, and guys, if you're not sure what I'm actually talking about, please do ask these questions because it's helpful and you'll be surprised how many other people are like, oh, I didn't think about that. I didn't link it together. So when I talk about a membrane, I'm talking about the fact that this is a slide I want to add in. I'm going to bring this in here. Where am I going? I want to bring it into this one. There we go. No, I want to go up. 1 second. There you go. So if I'm talking about membranes, I'm talking about this phospholipid bilayer, this separation of that, for example, extracellular fluid and the cytosol, the cytoplasm. Is a phospholipid bilayer? Yes, it is the phospholipid bilayer. And when you look inside, what you can see inside is that my cytosol here is colored yellow. I've done that on purpose because I want it to differentiate between here, there's a contrast, here to inside this section here. So this membrane-bound organelle, which is inside my cell, it's in my cytoplasm, my cytosol, it is a membrane-bound organelle, so it has made a compartment between the two. So what you've got is these phospholipids create a bilayer. So always use that term, phospholipid bilayer. And the fact that you've got the hydrophobic heads, I'll try that again, hydrophilic heads that are orientated on the outside and hydrophobic tails orientated on the inside. Now, I just did that and I want to make this very clear to you all. I did it. I knew I was trying to say, but I said the wrong thing. I got them mixed up. Now, I know I do that, and that's very much why I verbalize my answers to make sure that I am actually saying what I want to say and what I mean. I also do that when I'm writing. So if I'm sitting in the exam, I don't know if this is what you guys do, but when I'm sitting in an exam or if I'm doing something, what tends to happen is in my head, I'm thinking one thing, but I say or I write the opposite, even though I thought the right thing in my head. Now, I know that's a problem that I have. And so therefore, I'm always double checking my answers. And you'll be surprised how many exam papers I review that students like, oh, I didn't write that. And I'm like, but you did. That's all I see, what you have written. So always double check your answers as you're going. Okay, so plasma membrane acts as a barrier, obviously, and then you've got these membrane-bound organelles that are going to compartmentalize, basically make compartments of these structures inside. Um, do you know what? If it does happen to you, that's one thing. It's good that you know it because if you know it, you know to slow down. So I'm like all excited. The first time back, I'm like, "Blah, blah, blah," and it's coming out really quick. In the exam, you'll have that as well. You'll have those emotions that are going on, that adrenaline that's flowing that you need to just take a breath. Take a breath. Slow yourself down. Now, I know I do it because I get super excited and I love teaching and I'm very passionate about what I teach. And also, I'm very aware that I'm like, oh, I only want to give an hour. And so, therefore, I want to give as much as I can when I can. But in the exam, you have this time in the exam to be able to answer it. So, it's okay to slow yourself down. I'm time blind, so I have no idea if I talk for 5 minutes or 4 hours. Like it literally is oh, where did the time disappear to? So I have to, I don't have it now, but I when I'm teaching in a classroom, I have to have a watch or a clock on the wall that I'm constantly looking. I do it here. I'm constantly looking down at my computer screen to double check, right, what time is it? How long have I got? So being aware if you are somebody who is one struggling with formulating your answers or processing things, be aware how much time you should be setting for each of the different questions. Anyway, let's move on. So we had that our phagocyte was going to move towards the pathogen because of the chemicals. The key thing to note, that movement towards the pathogens is because of the cytoskeleton. So one of the roles of the cytoskeleton, again, module 2, is allowing the cell to move. So the phagocyte is going to be moving because of the cytoskeleton. The phagocyte is now going to engulf. Now, that engulfing, that projecting out, those pseudopodias, love that word, that are going to be projected out. So basically, the cytoskeleton is allowing it to put out these extensions that are going to surround the pathogen, and they are then going to come together like this. So 1, 2, come together. Now remember, that plasma membrane, that phospholipid bilayer is all the way around. So my hand comes together, they fuse together, and therefore what that can result in is, as you can see in my diagram, I've now got a membrane bound structure. Now, do not call this a membrane bound organelle. This has got a very specific name that you need to know. So we can see though that this structure here is not touching the plasma membrane anymore. And that is how we have done endocytosis. We have engulfed the pathogen and we've engulfed it into a phagosome. So by engulfing it into a phagosome, we now have got it in its own little compartment. And what's going to happen is this lysosome, again moved by the cytoskeleton, is going to fuse. See how it moved up here? If I go back, it's going to fuse and it's made of the same stuff. So it allows it to fuse. And what I then end up with is what's known as a phagolysosome. What's going to happen after that? Well, once they've fused together, that's going to release those enzymes, those hydrolytic enzymes that were held inside. That's going to release them out, and they're going to be able to do that digesting. So those lysozymes, which are the name of the enzymes which are inside the lysosomes, again, try not to get those words confused. So get it into your head. Lysosome is the membrane-bound organelle. Lysozyme is the enzyme inside the lysosome. You can see the diagrams if you switch over to that cannot be named. Oh, well done. So I'm just seeing, Will, your message. You got your identity offer. That's amazing. Well done. Oh, that is awesome. Really pleased for you. What's the best way to revise for biochemistry? Biochemistry is not actually that tough when it comes to biology. If you really truly understand your carbon atoms and the bonds that they make. Oh, guys, my phone's getting a bit too hot right now, so it may cut out on me for you guys. So sorry if it does. But yeah, best way to revise the biochemistry section is really just, I would say, don't focus on the, it sounds ridiculous, don't focus on the structures, but focus on the difficult parts of the biochemistry. So for example, when they are fusing together, what's the name of the bonds? That's something a lot of students struggle with, is naming the actual bond and knowing where the bond is created. The other thing they struggle with is the quantitative side of the biochemistry. So what tests are you going to do? So obviously you've got your GCSE level that you know it's Benedict's, but do you know how to describe it? Do you know how to use a colorimeter? Things like that. Yes, I am. In answer to your question, I am. Okay, back to this then. The lysozymes will digest and destroy the pathogen. What they'll also do is release cytokines. So these are these chemical signaling molecules, and they are to let the other cells know in the immune system that there is a pathogen that has been encountered. So more of them should come to that area. Excuse me. And then the other thing to note is just, if we're talking about a phagocyte, when somebody asked earlier, I haven't missed it. I have been looking. I've just been going as I went. You talked about APCs and humoral and cellular immunity. Please. So when we talk about antigen presenting cells, your APCs, you know, if you're still here, when we talk about them, what we're saying is those phagocytes are going to engulf and digest those pathogens, and those pathogens are going to have identifying parts to them called the antigens. So the pathogen's antigen is its identifying feature, and what the phagocyte will do is they will actually engulf the pathogen, digest it, and then stick a bit of it on the surface of its cell. Now, what they're doing is they are sticking it with what's known as their glycoprotein, remember, membrane, glycoprotein, part of the membrane structure. They're creating what's known as an MHC complex. So an MHC complex is basically a specific glycoprotein with the antigen being attached to, and that is where we call them now an antigen presenting cell. So when the macrophage presents the antigen on its surface, we've got this MHC complex, it has become an antigen presenting cell. And because it's become an antigen presenting cell, it can now stimulate cells such as your T helper cells, which can then stimulate your B cells. So the antigen presenting cell is the phagocyte, which has engulfed and digested and then presented that antigen onto the surface of the cell. And then just the other thing to know, this is positioning really badly because I've just copied these slides from my actual PowerPoint that I did for, let me move this down slightly. There we go. So the other thing to know is opsonins. So when you're talking about antibodies, an antibody could be an opsonin. One of its role, oh good, Julie, you are still there. It could be that it's an opsonin. So this antibody has actually bound to the pathogen. Now, what it's done when it binds by being specific, so antigens have a antigen binding site that is specific to the antigen. So when it binds the antigen, it sticks to it, it's tagging it. It's basically like sticking on a high-vis jacket that's saying, hey, they're not meant to be here, get them out of here. And that therefore makes them more recognizable to the phagocytes to then engulf and digest them. Way, there we go, finally. So that's just the other one to know on that front. But let's look at the humoral and the cell-mediated response. What am I looking for now? I need to move to switch things over. do I want? What do I want? What do I want? This one? Do I want that one? No. Ugh, I just opened the same one again. I didn't mean to do that. Go. Okay. No, I don't want you. I don't want to put you there. Here we go. Right, here we go. Oh, is my snottiness. Actually, that's really linked to this topic area, isn't it? So I've obviously picked up some pathogen, and that has resulted in me getting a immune response to try and fight off the pathogen. So my phagocytes are obviously fighting, but one of the things to note as well is that when we talk about your immune system, you've got your non-specific immune response, but you also have your first line of defense, things like your nasal passage having hairs and you having your goblet cells that are going to secrete mucus. And it sounds really lovely. The mucus is going to be there to trap and hold the pathogens or the dust or anything like that you've breathed in to prevent it getting into your airway. So that's the reason you have the mucus, to try and prevent that. But one of the things that's really interesting at the same time is that your nasal passage is really warm, and it therefore creates the perfect environment for pathogens to thrive if they're not trapped. So you're going to end up with this overproduction of the mucus, and it starts by just coming straight out. And for at the moment, I keep sneezing. You cut them. Do you know what? It's a funny one. When I first started teaching a class a couple of years ago, you saying that I've cut down your revision time, one of my students who came to my class, so I saw them 6 times a week, every week, and they were literally like, oh, what is this that you're doing to me? They were not used to my teaching at all. It was the first time I taught them, and they weren't sure, and I could tell because I can just sense they weren't sure. And then about 3 weeks in, they were like, oh my God, I don't actually have to revise at home now because I just get it because of the way you teach it. And I was like, oh, well, there we go. Yeah, and they got the A star, obviously. But yeah, I'm pleased that I'm helping you to cut down your revision time. Anyway, back to this. Phagocytes have the receptor. I went on a tangent about snot there. I'll come back to this. Phagocytes have got receptors, just like all different cells have got receptors that are important for the roles that they play. So that's the other thing to know. What is a receptor? So when we're looking at biological membranes and you're looking at that structure, the phospholipid bilayer, don't forget you have glycoproteins and glycolipids. They act as receptors, those receptors which will have a specific shape for whether it's a hormone or whether in this case it's the opsonins. So oh no, you need a new shoulder. It's not working. Um my when's my next lesson date time? So the actual lessons are starting next week for the group tuition. But I'll be back on here on a Wednesday again. Well, I've just realized you were on TikTok as well. Sorry, I missed you there. Right, let's carry on because I wanted to go on to I'm gonna need to move this stuff around. Yeah, okay, so if we look at B cells, B cells have unique receptors. Again, we're talking about receptors, and this is another thing I want to make you very aware of when you are doing your revision and when you're answering questions. So many students, for some reason, think that proteins can only be hormones. No, they can't. I'm lying. That's the opposite of what I'm trying to say to you. What they think is that the proteins can only be enzymes, and not anything else. Whereas a protein, for example, in this case, this is called a receptor. So try to read the question. What have they called it? Are they talking about enzymes? Are they talking about antibodies? Are they talking about receptors? This is a antibody which is attached to the membrane and therefore is a receptor. Hi, Tom. And so what you have to be aware of is that the wording that you use must be related to the question that they've asked. And so many students miss that. They just throw out, oh, it's an enzyme. No, it's not. Or it's an antibody. No, it's not. What is it? It is actually, oh, oh, no, hold on. I think my phone's going to cut out on my TikTok here. It's saying it will end because you're not actually doing anything. I am. Oh, no, what are you doing? Complete the verification. There is no verification. Oh, I just like my own life. That's hilarious. I don't know what's going on here, guys. So if it goes off for the TikTok, sorry, guys. I don't know what's going on. It's trying to get me to verify, but now it's saying it's not giving me anything to verify. So it's going to go off in about 4 minutes. So I'll keep going as much as I can, but if not, jump over to my other thing because I am here. Okay, so just be aware of that. Now, the other thing to know is when the antigen binds the receptor, that B cell will be able to process the antigen and present it on its surface. Is this protein the same as a protein that we consume by food? So, well, let's think about that. It's a really good question that you've just asked. Is this protein the same as the protein that you consume by the food that you eat? So the food that you eat, okay, has proteins in it. What are proteins? Proteins are made up of the chains of amino acids. So therefore, if there is a protein which is attached to the surface of the cell, that protein will have different amino acids, but it's a protein and therefore it's a chain of amino acids which is held together in the structure. So what I would say to you is, yeah, absolutely. Proteins are proteins. The sequence of the amino acids is what makes the proteins unique in their structure and what they're used for, but proteins are proteins. So therefore, whether you're eating the protein from the egg whites, if you like your eggs, or whether you're eating the proteins which are inside the plant-based foods, you're eating proteins. And we know that the universal code is because the amino acids that are coded for by the alleles, the genes, that's therefore connecting them together. It's the same stuff. So proteins are proteins. They are sequences of amino acids which are then folded and held together in a specific way. So it's important that you do understand, yes, it's connected. They will be different because they'll have different sequences of amino acids. However, that's how we use our understanding of the proteosomes or our amino acid sequences or our DNA sequences to look at the evolutionary link within the populations, within the organisms, within, for example, a domain or within a oh, my brain's lagging now. It's not just TikTok that's lagging, my brain's lagging. Within what am I trying to say? The taxons. If I watch all your videos on a topic and then do practice question, do you reckon I'll remember it all? Yeah, don't do that. And what I mean by don't do that, it's not about remembering it all. You don't have to remember it all. That's not what you've got to do. You've got to understand the fundamentals. If you can understand the fundamentals, if you can understand the key parts, and then you can apply them to whatever the question is that they throw at you, that is what's going to help you to maximize your grade. Just watching the videos, memorizing or learning just the video content is not going to help you. I promise you. I mean, absolutely watch all my videos because that just means that they'll get pushed out to more people and I can help more students, but that's not going to help you. What's going to help you is how you apply your knowledge to the questions. So lots of practice questions, absolutely. If you're doing practice questions, make sure that they are for your exam board and not other exam boards, unless, and here's the caveat, unless you are able to decipher whether you need to know that marking point or not. And that's one of the bits that I think some students struggle with. So I use in my group tuition sessions and in my one-to-ones, I use other exam board questions, but I know how to tweak them, how to ensure that they are just specific for what is needed for OCRA. And that's why, I mean, although I teach all the exam boards, I say teach, although I tutor all the exam boards, I made sure that I made my content only OCRA specific because there was nothing out there. There we go. TikTok's gone. It disconnected me. It didn't like me. So yeah, so that would be what I would say to you, Uni. But yes, watch my videos. Also, go and check out aiqchat.com if you haven't already, because that will also help you. So does that mean proteins make things in your body? Right. Okay. No. Be careful on that one. So proteins are made up of amino acids. Amino acids can actually have hydrophilic and hydrophobic regions to them. And what they do is they interact into a stable structure. So just be careful that it's not about something being acidic is about its ability to dissociate the hydrogen ions. So this is going back to GCSE chemistry for you now. So if something is actually able to dissociate a hydrogen ion, that the fully dissociated hydrogen ions means that it becomes acidic. And whether something is slightly acidic or whether it is strongly acidic, that's determined on whether there is full ionization or not. So just because amino acids have got a carboxylic acid group, what you have to remember is the amino acid coming together with amino acid. When you form that peptide bond, you are going to remove water. And because you're removing water, that therefore means that you're removing part of the region that could have actually had that hydrogen dissociate. So that's the bit to remember, that although it's an amino acid and the amino acid has a carboxylic acid group, when we talk about a protein, it's a long chain of amino acids that are held together by peptide bonds. We have, excuse me, removed water, and that therefore is going to make it not be acidic. Absolutely. And to be honest, guys, I think I'm going to finish now because I need to have a rest because I am still feeling this cold. I will be back on here tomorrow night. No, I won't. That's a complete lie. I don't know why I just said that. Next week, I will be back on here, not tomorrow night. I'll be back on here next week at about 8:30 for my next session. But if you would like, in the meantime, if you are looking for actually more dedicated, specific group tuition sessions for you, I am doing them on a Wednesday at 7:30. Oh, thank you. No, I'm not doing Mondays anymore for the YouTube side of things. I will do the Mondays and Wednesdays when we get closer to the exams. But right now, I need to focus on my income because basically I've got no, I haven't been paid for the last 6 weeks because obviously I've not been doing anything. So I need to focus on that, but I will still do a free live every Wednesday, like I have done. So it's dropping down to 1 day a week now on the Wednesday. But there is the group tuition offer groups for you. There is one-to-one tuition slots available if anyone is looking for that. But obviously there is also the revision platform AIQ Chat to go and look at if you are looking for just something, and it's only £5.99 that one now. We dropped it down to make it more accessible for more students, so do check it out. But thank you for my it kicked me out. I think my phone got too hot as well. The TikTok went, it was like bye. So yeah, I'm not worried about that too. to be honest. But thank you for joining me, guys, and have a good evening.